to Ricardo because everybody in this department knows Ricardo already. And so instead of really introducing him, I get to just tell you some awesome fun things about Ricardo, which is always the most fun part probably of the practices. So, Ricardo y toda su familia vienen de México y estaban allí también que Ricardo asistió a la universidad en matemáticas. En la Universidad Autónoma del Estado de Hidalgo. Y lo que es inusual sobre Ricardo es que es un vikingo mexicano. Cuando hace frío, con niebla y llueve y con viento, él se pone muy feliz. Okay, so probably everybody understood that, but I now switched to English because Ricardo then moved to the States. And I don't know about all the interim years, but he arrived at OSU in 2015, having done his master's degree in Mexico, where he comes from. And where he apparently enjoyed really cold weather in the highlands. Who knew? And so in 2015, he got to OSU and did a master's degree in applied mathematics here with Patrick Delenn here. Already interested in these disease dynamics at that time. And then in 2018, he enrolled in the PhD program here in integrative biology in my lab. And it was actually fantastic timing both for Ricardo and for the lab because we had just finished up a big field study on foot and mouth disease in African buffaloes. And the way that always works out is that you end up incredibly data-rich in analysis poor because you run out of time on a grant. After all the data is collected, then you have this mountain and you're like, now what? So here comes Ricardo right at the right time with good quantitative skills and an interest in modeling disease dynamics, when these data are available. So that was a lot of fun data for him to play with and a lot of analysis power for us to, push these projects further. And so Ricardo, during his time, has been co-advised by myself and Jan Medlock, but in actual fact, he's been co-advised by a trifecta of us, including Grimaldo. And that actually fits really well with Ricardo's personality. He is a really unusual mathematician who's incredibly social. So, enjoyed chatting with lots of faculty members. He also attended quite regularly Mark Novak's lab meetings because, you just need more intellectual stimulation while you're here. He loves going to conferences, presenting his work. I'm sure he'll love presenting it to you guys today as well. So let's see. What else? Ricardo's work has been really very interesting based on that mountain of data because he was able to push the modeling that we've done on disease dynamics several steps further, and he'll tell you about that in a minute. But I want to cede the stage here and let Jan and Bree also give you a little bit of a perspective on this time.
Yeah, so thanks, I'm Jan, and I think I want to keep it brief, keep my comments brief. So thanks to Anna and her great way of introducing Ricardo. I think I want to concentrate on saying, it's been, a lot has happened, and in particular, I got really close to Ricardo, and we thought to see him move on to bigger and better things, but happy for him and really proud of the work here with presentations.
I have nothing exciting to say other than Ricardo's awesome, and it's the first time I've seen him not carrying a pile of math textbooks.
You want to see my math book?
Oh, God. You're up, Ricardo.
Well, thank you for the lovely introductions. I can proceed and speak it in Spanish. My family is watching this, and actually, they did ask me if this was going to be translated in some type of way. I did mention that YouTube is available with a self-translation feature, but it doesn't pick my accent sometimes really well, so it might have a couple of cousins every now and then. They're non-intention, and they're mixed translations. Anyways, the title of the presentation that I'm presenting to you today is Foot and Mouth Disease, African Buffalo and the Immune System, a quest for the mechanisms behind the persistence of food and mouth disease in its referral host. As it was mentioned, I'm advised by Dr. Nayoles and Dr. Jan Medlock. The structure of the presentation, we're going to be having 5 specific points. We're going to introduce foot and mouth disease and African buffalo. We're going to tell you some of the knowledge about foot and mouth disease so far. Then we're going to discuss my first chapter of my dissertation, the inclusion of antibody dynamics for the epidemic process of foot and mouth disease. We're going to be talking then about the physiological characteristics, food availability, and other pathogens that can influence the drop of antibody protection on foot and mouth disease in African buffalo, and some of the conclusions so far of my dissertation. There is a third chapter that it is not included in this presentation, where we study the effects of the evolutionary mechanisms of foot and mouth disease as an RNA virus that allows for the persistence of foot and mouth disease. But for brevity of time, we want to just present some of the results of the dissertation. Some of the common terms and notations that you're going to be hearing on this talk is serotype, which is a distinct variation within a species of pathogen or among immune cells of different individuals. In particular, for foot and mouth disease, we're going to be talking about South African territories 1, South African territories 2, and South African territories 3, which are going to see color-coded with those 3 colors, blue, red, and purple. The importance of it is because antibodies that protect for one serotype, they're not protecting you for a different one, so they're completely different species from each other. We're going to be talking about serostatisis, which is whether or not an individual has detectable antibodies in their blood against a specific antigen. So seropositive means that they have a strong protection against a pathogen. Seronegative, that means that they don't have that immunity anymore. We're going to be talking about disease persistence, so the continuous presence of a disease on a group of individuals. We're going to be talking about R0. We went through the pandemic, you probably already know about this, the basic reproductive numbers, so the average number of new infections transmitted by an individual during the entire infectious period. We're going to be also talking about hazard rates, which just think about the instantaneous failure rate of maintaining serostatuses. And throughout the presentations, we're going to be looking at this symbol, this little drawing of a buffalo. It means that the data presented to you or the results presented to you, they come directly from data from the cohort study. And if you see this little symbol, that means that those results, they are from simulated data, right? So now that we have all those common terms and notations, we're going to be talking about foot and mouth disease. As I mentioned, it's a single expanded RNA virus, which is usually transmitted directly or by fomites. Think about spit, sneezes, and whatever. It's a respiratory disease. It has a global distribution and infects individuals. So the graph right here, it shows where foot and mouth disease is spreading, except on all these green territories. So because there are regions of the world that have foot and mouth disease free, it does have an impact when trading of cattle or different ungulates that they have hoofs are passing between countries where they who have foot and mouth disease to the ones that are foot and mouth disease free. If you were alive in 2001, you probably remember the big epidemic of foot and mouth disease in the United Kingdom that basically decimated the population of different farms. Foot and mouth disease is one of the most contagious infections known to man. It's actually second just to measles. So basic reproductive number of different diseases are estimated here, and then foot and mouth disease is just a little bit behind of measles. And as I mentioned, because foot and mouth disease affects cattle, and there are regions where they are foot and mouth disease free, it's one of the most trade restrictive livestock disease, especially for those developing countries that are not foot and mouth disease free. This is one of the notices on the 2001 United Kingdom epidemic, where some regions they have to be completely isolated in order to deal with the epidemic of foot and mouth disease that they face. In the sub-Saharan region, African buffalo, Sincerus café, is the reservoir host. That means it's an individual and a specific host that carries the disease. They don't present specific symptoms, and they just are continuously infected. In foot and mouth disease, usually will cause mild lesions. In buffalo, they are typically none. In this picture, we have lesions that they can cause on cattle. Usually, you don't see this type of lesions on buffaloes. They're very resilient towards this particular pathogen. As I mentioned, there are 3 different serotypes: South African territories 1, 2, and 3, which are located in different regions of the African continent. And it's really efficient by transmitting between the 3 ranges of cattle and the buffaloes. And usually all buffaloes are going to be seropositive for all 3 different serotypes by 1 or 2 years of age, right? So what does it mean? Basically, what we're facing is that buffaloes, we have herds that they have a wide range of population size. They can be between 100 individuals to thousands of individuals. And then you have foot and mouth disease, which is spread quickly. So the exposure time usually is between half a day, and the infectious period is usually about a week. Foot and mouth disease is not deadly for the buffalo, and then they recover. So usually your epidemiological knowledge will tell you that if you have small populations of fast-spreading disease, that they recover and it's not deadly, it should just pass through the entire population, everybody recovers, and the disease die out. But it persists. So that's a big problem for and a really interesting problem for foot and mouth disease for us to study. And particularly when we're talking about wild populations, it can be challenging because there's no way of, isolate them or like capture everybody, vaccinate everybody. Mechanisms of control that usually can be done on cattle, they are not necessarily applicable for wild populations. So we want to understand what are actually driving the persistence for this fastest spreading pathogen on this wild population. So the goal of this dissertation is explore how different within host mechanisms and between interactions can scale up for the persistence of foot and mouth disease on this wild population. And again, some evolutionary mechanisms can facilitate the persistence of this pathogen by just facilitating the reinfection with previously recovered individuals. The way that we do this, since population sizes are really small and they have variability and they face different factors, we utilize stochastic modeling in order to assess the entire epidemic process. So the way that we denote this is an MSAR model, which is a maternal immune, susceptible, exposed, infected, and recovered model. We're going to go in detail more and for the model that we're using. But these were the specific models that were reported on a science paper by the Julius Lab in 2021. So again, you have maternal immune that are going to be passively susceptible, exposed, infected, and recovered, or susceptible, infected, exposed, and then they have chronic or chronically infected individuals that they eventually become recovered. The transitions on all the different stages of the epidemic process, they are modeled through waiting time distributions. In all these models, the death was age dependent and the birth was time dependent. And that's a constant behavior that we have for our model. Now, I've been talking about a little bit the model, about the disease, about the host. We need to find the study. That was what it was done in the previous set of studies. So you can see that the different serotypes of interest are distributed on the entire African continent, but in particular, they are all located on the northeast region of South Africa, in Kruger National Park. That makes it a really good area for study. This is in the area between close to Mozambique and Zambia. So that's where the cohort study was located. There was a herd of 60 to 70 buffaloes. They were captured every 2 to 3 months for 3 years, and then we were monitoring foot and mouth disease. So the original hypothesis was that maybe foot and mouth disease was a calf-to-calf transmission. If you have kids, you probably have heard of hand, foot, mouth disease, which is the version that we have for humans. It's a similar virus. It's a completely different species. As pathogens, they're differently, but similar idea. So the original hypothesis of calf-to-calf transmission is what maybe foot and mouth disease was transmitted like a typical Chagas infection with annual epidemics. So that meaning that the latest born cows of a specific cohort were able to infect the earliest born cows of the next cohort. The other hypothesis that was done on this study in 2021 was that maybe foot and mouth disease, since foot and mouth disease persists in buffalo for months, years on the palate tonsils, maybe the carriers, the constantly infected individuals were sparking those annual calf epidemics. But we needed to know when or how often the chronically infected individuals were able to become then infectious again. So in the question of can foot and mouth disease persist via calf-to-calf transmission, we found out through the different stochastic models that because FMDV has a massive inversion persistence, the virus cannot persist by acute transmission in cows. So calf-to-calf transmission doesn't fully explain the persistence for foot and mouth disease. The carrier transmission, well, it did show some robust persistence for foot and mouth disease. So this is the total number of infected individuals And you can see that it never died out, average of about like 10,000 simulations. But then there was no persistence for SAT2, since all the different simulations eventually died out. And for SAT3, there was some persistence of about 60%, but the variability also affected the different set of results. Now, all this is introduction material to just 10% of you what my actual work is about, right? Antibody rises usually after the first primary infection, and that's expected to happen. So individuals should have high antibody levels consistently, they just recover from reinfection, or they just expect to have a waning decrease of antibody titers. So the recovered individuals that eventually they become susceptible. Those are the expected behaviors that we're going to one expects to see on this type of diseases. Buffalo usually lose their protective antibody titers. That was, and that allowed for the variation of protection to be variable among the different individuals. So just the estimations for the duration of immune protection, it was different among the different serotypes on the individuals on the study. So that became an issue. It was a little bit counterintuitive to what one would expect of this type of diseases. And because of this, all susceptible animals were in all different age classes. As I mentioned, most animals are basically seropositive or have been infected by fundamental disease by 1 to 2 years of age, but then individuals also became susceptible back and forward in all different age classes. That's not normal, right? So that kind of shows that there was something else behind driving the persistence. My first chapter, a novel mechanism for persistence in highly contagious pathogen, the unstable antibody protection against foot-and-mouth disease in African buffalo. So we notice antibody variation on the study, right? Different individuals, they become seronegative to then become seropositive, probably after an infection, or just the good food availability, or they just become, they were always seronegative, so they were always susceptible, or they were like this one, where they were like passing to be seropositive to seronegative and back and forward. And because all these different antibody dynamics, right, it kind of leads us to maybe that antibody variation was helping with the continuous infections of foot-and-mouth disease on those animals. We also noticed how across the study, the proportion of individuals that they have high antibody levels was highly variable between the different serotypes. So more research question was that how does the variability of antibody protection influence the epidemic dynamics for foot-and-mouth disease in African buffalo? And if the drop of antibody protection can explain the persistence for foot-and-mouth disease. So knowing that antibodies can drop and antibodies can be gained, we wanted to know how fast or at what rate those antibodies were being dropped. So what was from the probability that an animal has high antibody levels, can drop, at what rate can that probability drop that they have low antibody levels? So the hazard rate for the antibody dynamics. We estimated those hazard rates for the study. So those having the hazard loss rate throughout the entire study for hazard loss. So how fast they were losing antibodies. For the gain, since the gain of antibodies can be because of infection, or can be because of the different conditions on the animal, like nutrition or different stress factors, even infection from other pathogens, that variation changed at the beginning of the study from the end of the study. So we accounted for that. So we wanted to know what was the rate at the beginning of the study, the rate at the end of the study, and maybe how did that vary throughout the study. So our model. The model was an extension from the work on 2021. Again, we have a maternal immune class. So the duration of maternal immunity is about 135 days. Individuals that they have waned the maternal immunity, they become susceptible. Maternal immunity, they can only pass from the first colostrum from individuals that they have recovered from the disease, or they are currently carrying the disease. They have some type of level of antibodies on their system. Susceptible animals, they can then become infected, different rates of transmission, either from infectious individuals or from chronically infected individuals are passed on this for the different serotypes. And those are number of individuals per day. There is an exposure class. So once that they have an infection occur, there is a small period of time where they are still not getting infectious, they're just exposed to the disease, usually lasting between half a day to 3 days. And then they become infectious, which they are infectious for between about 4 days to a week. There is a chance that those infectious individuals, they become chronically infected individuals, so they become carriers, and the carrier period can vary between 243 days to 174 days for different serotypes. And then they can also, individuals that they were infectious, they could fully recover from the disease, those having high level of antibodies on their bloodstream, which they can then lose at different rates. And then those lost immune or immunocompromised, if you want to think about it like that, individuals, they can then regain those antibodies at a specific different rates of change. Now, one big assumption of the model is that those individuals, they are immunocompromised, they can become exposed again, not at the same level as fully susceptible individuals. They do have still some resistance, but it's not perfect. They're not completely immune. And because they don't have enough antibodies on their bloodstream, they cannot pass that maternal immunity. They cannot pass that immunity to their calves. So then they produce newly susceptible individuals after they give birth. So this is the model that we were studying now. So again, we utilize stochastic simulations. If you have done any type of stochastic simulations, you know that you usually have a set of initial conditions. In order to find those initial conditions, we have a deterministic version of the system. We find the equilibrium for an endemic state because we are not interested on the beginning of a disease. We're interested on the scenario where the disease is already there, it's already endemic, perfectly controllable, but not controllable, already is well established on the system. And then we use that as the initial conditions for this stochastic process. So we needed some sample of the parameters. I mentioned the different hazards that we were studying from the actual study. Then we do Monte Carlo sampling to basically get more samples of all those different rates for the loss of antibodies and the gain of antibodies. We get about 20,000 samples of each one of those. And then we use those to then run a stochastic simulation. And just one stochastic simulation, in this case for SAT2. We do this through Gillespie algorithm. So then you can see that you have already well-established recovered set of individuals that eventually they wane down, since those recovered individuals that are going to go to the low immunity class or the immunocompromised class. And those immunocompromised class, then they can become reinfected. The susceptible class that started somewhat high completely goes down and then just continues to oscillate because births they can keep occurring, or the maternal immunity that eventually they went down. Then the exposure is always constant, and then the chronically infected, they just keep growing. So what we observe here, in a period of 10 years, there is always at least one infectious individual or an individual that carries the disease, either from the infectious class, the exposed class, or the chronically infected class. So that's what we observed in one simulation. But that's one simulation. Maybe it's not giving you everything about it. So what we do then is we run 10,000 simulations for each one of these serotypes. They're completely independent from each other. So if you're looking at one of the lines over here, it's just one run like the one I showed you before for each one of the different serotypes. We account the total number of infected individuals. So those that they were infectious, those ones that were exposed, and the ones that were chronically infected. We add them all over, and then we see how many of them they were prevailing at every single point in time for a period of 10 years. After 10,000 simulations for each one of the different serotypes, we are able to see that SAT1 still persists. SAT3, stabilizes better. So then we have the persistence for the serotype SAT3. But for SAT2, we see a few of the different runs eventually died out. So only 99.8% of those 10,000 simulations were able to have full persistence for a period of 10 years. Some of them, they died around 6 or 7 years, right? 10 years was chosen to be a measure of like, this is a good enough long period to have a new set of cohorts, maybe joining the current cohort of study or a current population. So it's a good measure for disease persistence, right? Periods could be shorter time, perhaps, but 10 years is already long enough that it's about half the lifespan of those animals. So eventually any random animal is going to see this disease and is going to be infected by it, recover it, and eventually see it in other individuals. This is the main result for first chapter. We saw that just including the antibody dynamics explains the persistence for one of the serotypes, along with the chronically infected individuals, as it was with the study in 2021. SAT2 still has some issues, but there are more ways that individuals can be losing antibodies. We're going to be discussing that in a little bit. Since we were introducing a bunch of new parameters, we wanted to know if the results that we're observing in the model, how sensible are those to the variability of the different parameters that we're introducing. So since we introduced the immunocompromised state or the loss immunity state, we wanted to see if varying the susceptibility of that class modified heavily the set of results. So think about it in this way. Susceptibility of 0 is that loss immunity state is a completely fully immune state. So it's the same as the recovery class. A susceptibility of 1, that's the baseline susceptibility that we set up for this class. So the persistence for the susceptibility at 0 in all the 3 serotypes, it did match the results on the paper in 2021. It's good. It'll be contradicting previous results. Once that the susceptibility increases, well, we still see persistence on SAT1. The persistence on SAT3 immediately rose, reaching persistence by a susceptibility of about 20%. And in SAT2, the susceptibility just grows slowly, monotonically, but still slowly. We also try to see how the model was sensible with respect to the population sizes. As I mentioned, the herd of buffaloes, they can run between 100 individuals to thousands of individuals, or based on all the different simulations was 1,000 individuals. So then we did the same of varying the population size when having full susceptibility on the low immunity state, and see how, as the population grows, the persistence eventually catch up. So the persistence on SAT1 catch up immediately. That was good for us to see right away. The persistence on SAT3 grow slightly slow, but it was still present by a really small number or small population size. And the persistence for SAT3, it did require to have some high numbers on those populations or those herds in order to have beyond the 50% persistence. Now, we have sensitivity with respect of the population size and sensitivity with respect of the susceptibility. Let's do both, right? So then you see across the different population sizes and all different versions of the susceptibility, we see what are the regions that we have, enough or high percentage of persistence after 10 years. So SAT1 showed us that, for really small population sizes with really high, with really small population sizes and some high susceptibility, we do show a 99% persistence, even 50% persistence. It happened even for small population sizes and small susceptibility. SAT3 was starting to show 50% of persistence, in most of the regions of the study. And SAT2 was the one that was still kind of evading that persistence mechanism. So some work needs to be done for SAT2 in order to fully explain the persistence. But again, in this case, we're just accounting the loss of antibodies just to have that other mechanisms that they can be affecting the antibody drug protection. Through the same sensitivity analysis, we were able to see which one of the different parameters were more influential. The most influential were all associated to the transmission rates of either the chronically infected individuals or the acute transmission rates, or the durations of the infectious class, not the loss immunity class, which is good. That means that our new set of parameters that we're introducing to the model, they're not going to vary the set of results, significantly. which means that even if we were making some error on the estimations, there's still some leeway, let's call it like that. So in this first chapter, we saw how the different serotypes, they have drastically different epidemic dynamics. The inclusion of that immunocompromised state stabilized the process or the persistence for SAT3, but not for SAT2. Again, still has some issues. At the beginning of the study, the SAT1 antibodies, they return to the protective titers faster after a drop than those on SAT2 and SAT3. But at the end, the antibody gain range of all three serotypes seem to have converged about the same region. So that possibly represents that there is some other explanation of why the antibody races are occurring and also why the antibody drops are occurring, which actually gives us a segue to my second chapter, right? Understanding those antibody dynamics in the wild. So as I mentioned, there are more factors that they can be affecting the drop of antibody protection. It's not only like time, it also can be that these are animals that are in the wild, food availability is an issue. Maintaining a good, strong immune system means in a trade-off with like actual resources. So our research questions for this chapter were, how general is this finding that antibody titers drop quickly across different pathogens? That do antibodies against different pathogens tend to drop together or opposite to each other? And what are other physiological and host factors that can promote the antibody drops? So the reason why we ask that is because African buffalo can be affected by other pathogens, not only foot-and-mouth disease. If you were here a couple of years ago, there was a research colleague that came and talked to us about how African buffalo, they actually even get anthrax, which is a really nasty infectious disease. They also get other different respiratory pathogens. So because they are affected by several pathogens, the antibody drop for foot-and-mouth disease could also be driven because of other infections from other pathogens. So we expect that several stressors might be playing the role on the maintenance of antibody titers of a given pathogen. That again can include the infections, the reproductive cycles, the fat reserves that the individuals have, and the forage availability and quality. Those are the processes and constraints that can mediate the metabolic allocation for the immunity. So the production of antibodies, right? How good your diet, how the stress with your environment can influence how good of an immune system. If you're really stressed, you're pretty easy to get sick. Like I almost did yesterday. So some of the measures of interest for this study was that, well, we wanted to see the sex of the individuals on the study. So that was done through a binary matching, 1 for female, 0 for male. The age in years. So there were a few cows born during the study. So the range of ages goes from basically 0, it's like a couple of days old, to about 20 years of age, with an average of 6 years of age. The average body condition, which is a categorical variable, usually the average scale goes from 1 to 5, where 1 is really poor condition and 5 is really excellent condition. And that was done through palpation of ribs, spine, hips, and the base of the tail. So already good enough established measures for the average body condition for these individuals. We study the levels of albumin, which, if you don't know, is a protein produced of the liver, and usually is a good proxy for the nutrition and the hydration status of an individual. We look at haptoglobin, which is another protein of the liver that is prevents oxidative damage, but is also a good marker for inflammation. We look at hematocrit, which is a percentage of volume of red cells on a sample at the different captures. We also look at the non-esterified fatty acids, which are called NIFAs. From now on, I just say NIFAs. This is a mouthful. I'm glad that I was able to do it in just one go, which are just those adipose fat tissues that they're released as a response from starvation. So basically, it's a good measure to know if they're malnourished. There were different dietary conditions in the study. So individuals faced three different dietary ratings. They had restricted food availability because there was a drought during the study. They had the food availability during wild conditions, and then they have a Ka feeding rate. This gives us a good measure or a proxy for population level nutrition. And we also look at the fecal chlorophyll, which is the amount of chlorophyll that were present on fecal samples when they were captured. So we look at their poop. That gives us a good proxy for individual level nutrition. In the study, we also have, we witnessed antibody presence for different pathogens. The ones that we were interested on were adenovirus type 3, bovine herpes virus, bovine respiratory syncytial virus, bovine viral diarrhea, Mycemia hemolytica, Mycoplasma bovis, parainfluenza 3, and of course, Fluenza disease. All of these different pathogens, they are caused by different pathogenic agents, either viruses or bacteria, that affect different parts of the body. They were assessed with different types of testing. And in particularly, we were interested on the drop of antibody protection for each one of those pathogens, because they come from different tests. The measure or the way of how we assess that antibody drop protection was happening had to be done differently. For those that they were done with indirect ELISA, if the test have some presence of antibody and then went to 0, so no presence of antibody binding, that was a drop of antibody protection. For those that were with a competitive ELISA, it was if they went from above 50% binding on the test to below 50% binding. And Fluenza disease, since it's an LPD ELISA, it was again the measure of 1.7 titer to below 1.7 titer. Furthermore, three of these pathogens, adenovirus type 3, parainfluenza 3, and Mycemia hemolytica, usually they are observed to be appearing at the same time, often in cattle population and in buffalo as well, and over different mammals that are affected on. So we're expecting that maybe they behave in some type of similar way. The number of antibody drop events, or the proportion of antibody drop events that they were happening for each one of the different pathogens, range between 12% to like 1%, something like that. Different sample sizes were present for the different pathogens. This is just a good report, just to know more or less how frequent this drop of antibody protection were occurring. In a similar fashion as we did with the Fluenza disease, we estimated at what rate they're losing that antibody protection during the entire study and how often they're gaining. But again, gaining of antibody protection can be because of different reasons. So we're not going to focus on explaining the antibody gain, since there is no one unique way that we can explain it, because they could be because of an infection, or they can be because of the physiological factors as we are interested in studying. But the drop of antibodies definitely it is mainly driven by the physiological factors. So we're going to be looking mostly at just the antibody loss. But for good measure, we also look at the antibody gain. Again, AD3, MH3, and PA3, they usually occur together. So we're interested in seeing how big are different rates of losing those antibody protections, which we can see they're similar enough, just with wide ranges of variability on the study. For MB, BVDV, VRSV, and BHV, we also saw different patterns on at what rate they're losing antibodies. Now, we were interested on, are the drop-ins happening at the same time, or are they happening at different times? What is going on? So we look at if the drop of antibodies were correlated or not. So through Pearson correlation, we were able to see that for the trifecta of respiratory pathogens, PI3, AD3, and VHV, when one of the antibody protection dropped, it dropped for the other two, which is good to know. It was surprising for us to see that. MH joining that group, they have a really strong correlation on their losing antibodies at the same time. The 3 serotypes for Fluenza disease, they also show some positive correlation on the antibody drop, and they also were positive, or they were somewhat synchronized with the trifecta of respiratory pathogens, which also was interesting to see. But BBDV, NMB, and VRSV, they were like, the black sheep of the family. They were completely behaving their own way and not even having the same type of correlation between the even similarly enough pathogens, which again, it was interesting to see for us in this study. Since we're measuring the presence of antibody drop protection or the absence of presence of antibody drug protection. So we have a binary measure. The best way of how to assess, what are the different physiological factors that they're affecting that drop of antibody protection is through a generalized linear model. Nevertheless, we have small population sizes, so a Bayesian generalized linear mixed model is the best way for us to assess that. So that's the model. So we're looking at the logic of antibody drop, and we're seeing what are different rates among the different characteristics of the sex, age, body condition, fecal chlorophyll, the dietary regime, hematocrit, haptoglobin, in NIFA, and allowing for some glucophera, and the random effect coming from the specific and that we're looking at. This is basically one of the few questions we have on the slides that I don't know, I was happy to put that. Since it's a Bayesian framework, we can look at the probability of direction. So highlighted are those that are above a 95% probability of direction. That tells us that for these specific measures on those specific pathogens, those show some influence. Nevertheless, the probability of direction only tells you, is it going to be influential? It doesn't tell you how. We're going to be looking at how these are being affected. But just from, taking a look, the type of diet was, influential in most of those pathogens. The haptoglobin, which again, one of the proteins that is associated with nutrition, it was also somewhat influential in some of the diseases. NIFA, which again is a measure for starvation, some influence on promoting or restricting the antibody drop of protection, and the same with the body condition, right? So those we observed that were happening among all the different pathogens. Sex just affected a few of the different pathogens, and also the age. But basically from this part, we were able to see that nutrition, the type of nutrition that the animals have, the fat reserves, and the protein reserves that they have in order to produce antibodies did affect the drop of antibody protection, or the lack of drop of antibody protection. So let's focus on the MH, PI3, 83, and B3. Again, the respiratory trifecta and MH. So we see that the restricted diet, so the amount of food that they were having during the drought was negatively affecting the drop of antibody protection. This is a little bit of a surprising set of results, but at the same time, this is compared to when they were fed on hay, and also keeping in mind that nutrition has some delay. So maybe we showed that the restricted diet was less prone to promote the antibody drop protection, but it also could be because we might detect restricted food after we were feeding them hay. So it's that change of the one that we can assess. The increase on NIFAs, so the fats, the improvement on their body condition, basically was also preventing the drop of antibody protection. The body condition definitely was preventing the antibody drop. So again, 0 is a measure of like if there is no change, and if it goes to the left-hand side of that vertical dash line, that means that it's preventing the antibody drop protection. If it goes on the right, it's enhancing the antibody drop protection. So for 83, PhD, and PA3, so that trifecta, the body condition is affecting the antibody drop protection. So if they are better fed, they are less likely to drop antibodies. And haptoglobin also shows some promises for BHB and MH, not too significant for the other two, mainly because if these intervals of error, they intersect that line, that means that there is not much of a strong evidence for it. And they match with the probability of direction that we show on the previous slide. For the food and mouth disease virus serotypes, we saw that haptoglobin was influential for 2 of those serotypes. The type of diet was also influential, and the body condition affected SAT1 and 2. Sex affected somewhat SAT2 on, basically female buffaloes. They were having more antibody drug protection with respect of males. The fecal crop also was preventing the antibody drug protection. And then MB, VRZ, and BBDB were actually showing the opposite behavior to the ones on the trifecta, which again, we were able to somewhat expect that when we're looking at that correlation among the different antibody drops. And again, all of this is coming from the data and also, some of the simulations, because the Bayesian approach does require to do some Monte Carlo sampling, so then there's some simulation you have to. So in this chapter 2, the evidence that we have is that there's a wide variation on antibody response longevity across the different pathogens. They can range between weeks to years. The drop of antibody protections seems to be synchronized for and driven by the host condition and nutritional factors across the different acute respiratory pathogens. So that trifecta of 83, EI3, and BHV, and also MH, because that happened as well, which have been previously shown to occur in individual buffalo. I mean, the presence of those 3 respiratory pathogens. The drop of antibody titers against the 3 serotypes of lung disease were also correlated in this group, although they were a little bit weaker. The antibody against the other 3 chronic pathogens, so BBDB, MV, and VRSV, show a slightly like a really negative association with the trifecta of respiratory pathogens, which was surprising for us to see. And so we saw that the host and environmental or nutritional drivers were similar among what drove the antibody drug protection for all those different pathogens. And the antibody dynamics, again, of natural host populations are still poorly understood, but we have seen, because of my previous chapter, how they can profoundly affect the disease dynamics. So understanding what exactly is preventing or enhancing the drop of antibody protection is a open field of study, particularly because it's not easy to capture the signals. So in whole conclusion in my dissertation, so we have a combination of experimental and cohort studies, which allows us to facilitate the estimation of realistic parameters for epidemic modeling. The drop of antibody protection can be affected by the different host rates and also their dietary conditions. We saw that similar pathogens behave in similar ways and by the different host and nutritional conditions. In this dissertation, basically, we viewed some of the mechanisms that they can affect or they can explain the persistence of food and disease. In chapter 1, the loss of antibody protection. We saw also the presence of other antibody pathogens, the condition of the animal, and again, the evolution of the pathogen. Those are all my conclusions for this dissertation. Now, nevertheless, if I've been mentioning, my dissertation and stuff, that's not true. Right? There are some acknowledgements to do. So I'd like to thank some people. This is usually the part where we get to talk with reply the introduction that we were given. So first of all, I would like to acknowledge and thank deeply my advisors, Anna and Jan. They gave me a lot of opportunities, and they have had a lot of patience throughout these years to not only deal with a lot of personal stuff. They have guided me through both the scientific career and also in a personal way. They have offered me multiple ways. I do consider them more than my advisors and my friends. I want to thank my committees, current and past, since changes happen. Academia, they can sometimes people, move between the different positions. So Dr. Bichler, Dr. Patel, Dr. Sher, Dr. Deal, I do thank you for all your patience and also, conversations that we have had. When sometimes I seem to disappear, but I'm still here. I'm just still working, so I'm trying to crunch those numbers because those simulations sometimes take forever. The different institutions that actually facilitated me to have these studies, again, Oregon State gave me the chance to be able to reach different places the Society for Industrial and Applied Mathematics and the American Mathematical Society, gave me lots of opportunities to present my work in different settings. I want to thank all the people on the South African national parks and Kruger national parks, because without their help, I would basically wouldn't have been able to do all this, because they are the ones that they captured those animals. I did not touch a single buffalo in this study. There were several instructors that affected or guided me as a teacher. I do want to thank you all. The pictures I could find. So thank you. Different parts on life, and Dr. Case as well. And yeah, I want to thank people in the office, because they do run this department. They keep it running, and they did help me a lot as I was trying to schedule, handle paperwork. And if you know me, I hate paperwork, but it's partly because of cultural conditions, since paperwork in the country is a pain in the butt. So they did help me to keep it all together. I want to thank the Jonas Lab, all the current past members, which they have been a lot of people that they have influenced me on discussing the scientific process of infectious disease of wildlife. Shout outs to Dr. Devan Song that actually helped me to also keep my writing together, because I'm not good at writing at all. But then she will be questioning me every now and then and preventing me or challenge me if I don't complete my writing. I cannot get to eat yummy food or spicy food, which, if you're from Mexico, that's a cardinal sin. Again, more people from the Jonas Lab, that they, have been a pleasure to be discussing and passing, different tips, recipes, and whatnot. Also, the Novak and the Lyra Lab, they have given me the chance to talk with other people that are on my field, have good discussions in the scientific realm. Of course, I do not have all the pictures of both labs. I could not find them all. There's so much stalking that you can do online, that you can do. Organizations, right? I want to thank CBE as our union. They gave me a lot of interactions with other graduate students on other programs, see their perspectives, in the STEM fields. They often we see things on a specific way, and then we see that how things are not the same on other fields. And also IMUS, the inherits in STEM, since sometimes being of a minority in the STEM field is not easy. So having a place on where to discuss this type of challenges that we could be facing, it also helped a lot. I want to thank my cohort, wherever they are. Some of them, I know they're busy right now. Most of them, all of them have already finished. Collaborators around the world, around the campus, they have also given me a bunch of advice and experience, wealth of knowledge. Friends from all the different areas that they always give me, also keep me going, keep me sane, as much as I can be said. My family, not only back at home in Mexico, here with my family in the United States. Not only family on the humankind, but also on the fur kind. They do keep you going and give you sometimes a reason to just get up. And just that. Well, that's all. We have questions.
Well, we think we have time for a couple of questions, but I wanted to interject really briefly before Ricardo takes those, that we will be gathering at our house, 5:36, and there'll be food and there'll be drinks, there'll be company. Sarah has sent out, I think, to the whole department, the invite with the address on it. But if you don't know the address, speak to anyone from our lab group. What questions?
No questions are also okay. Yes.
Just a couple very basic questions about how you did your study in terms of the immunology and maybe a little bit about detecting the virus itself. So, this is such a big problem. It's so great to have studies like this.
Yeah.
And it must have been pretty rough to go out and lasso those Cape buffalo and take swabs out of their mouths. I mean, that's not an easy job. But in the ELISA that you did, I assume you're assaying for neutralizing antibody, right? An antibody that's been previously shown, at least experimentally, to neutralize the virus.
Yes.
That's correct?
Yeah. Yeah. Also, I should have start with that disclaimer, and I think it was mentioned. I did not do the field study. I did not do the collection, but yes, that's what was done. The LPD ELISA for coronal disease is already established, or a test established for cattle already, and or the detection. And it was also specifically made for this particular study. So joining specific antigens for the 3 different serotypes. So it already detected that and detected the level of antibody titers on those blood samples. Yes.
And but there were no vaccinated animals included as controls in the study overall. You guys don't, because there are vaccines, right? Of
Yes.
some kind. I mean, they're serotype specific, but I mean, I don't know how viable they really are.
So that is true. There is a sign for vaccines for controlling the foot and mouth disease epidemic. That's one of the works that the PERBAC Institute, which is one of the collaborators in this study, that's the work that they do. Estimating what the design of those vaccines are still some of the work that still they try to do. They try to vaccinate as many of those animals as possible. For this study, my recollection is that they weren't vaccinated. So
Not vaccinated.
yeah, they were just wild population.
And in terms of the virus persistence that you were talking about earlier in your talk, is that based on immunologic data, or did you actually have any evidence of virus from the lesions or something? I'm just kind of wondering if the virus was, how you monitored live virus infection.
So
you know what I mean?
yeah, so there's some swabbing that was done to measure the presence of the pathogen on the buffaloes. Yes, when I was talking about the persistence here, it was from the data we estimated. Okay, there is a threshold before they have high antibodies, they have low antibodies. That comes from the literature and also from the data for those levels was directly from the swabbing and blood samples, the test detection. Our measure of persistence was once that we put that all in a model, we see how many individuals are infected or have presence of the virus after 10 years, right? And the problem of persistence on food analysis in the wild, that exists. They often capture buffaloes just as survey, just to measure, to see how healthy they are. And then as part of the protocol, they swab them, see if they have been infected and they have the presence of the pathogen. So they assess, okay, these are still infected or they have some presence of foot disease for this herd that we found in the middle of the wild. So that's not a question for us of like how if there is persistence. We know persistent it is, we don't know why it's causing it or how it's still maintaining itself. So I was part of the study. Yes.
Well, 2 questions as well. In your second chapter, how did you deal with individuals that flipped whether they were immune or not? Because it seemed like your regression is binary. Either it is or it isn't, but it didn't have a temporal component to it in that model.
So the part of there is the temporal component comes from just the different measures. The capture time is not put on the model directly, it's just with the animal ID. Yeah.
So it's ID by time.
Yeah.
Okay, thank you. And then if I can ask the second methodological question, this is more about the first chapter,
Uh-huh.
slide 22. You said that right before you said that you did the deterministic model first.
Yes.
That is initial conditions for the stochastic model.
Yes.
Why then do you have transient dynamics in the stochastic model? Why did, I think it was the susceptible
Mhm.
class, why did they decline? If you were at steady state already?
You're in a steady state for this deterministic model, right? So you're accounting for age of individuals, time from infection. This is the overall behavior, and then we put it on the stochastic model. So then there is new individuals that they might be emerging. The population changes eventually. They still face. There is some probability that they might lose antibody protection, or they might,
So there's a new steady state that emerges, an alternative, not an alternative, just a new steady state that emerges when you add stochasticity.
Right. But also, there is a difference between an steady state from a deterministic system and behavior on a stochastic system. So then think about it in this way, there are rates that are changing values in just the deterministic system. So take the steady state of a deterministic system, and then you change one of the rates values. The steady state should change, right? The steady states are dependent from the parameter values. So if the parameters are changing at any moment, which they do in the stochastic version, because we have a distribution set of values for each one of the parameters, the behavior is going to be slightly different. I don't know if I'm explaining that well.
Yeah, I think I understand what you're saying.
Yeah.
Doesn't seem slightly different, it seems very different. And if you're, I'll ask you more later.
Yeah.
I've got time.
Yes.
I also have two questions, if I may,
Sure.
but more around like the interpretation, I think, of some of the results. So you showed that the restricted diet increased susceptibility, is that correct, for certain disease types?
Yeah, so the diet regimen is a little bit tricky to do because we have individuals that we haven't registered of, like, at this period of time they had restricted diet, or they were just having food availability through a drought, and some of them were compared to hay. The restricted diet measure that I was showing is, think about it, it's like the baseline is the hay against the restricted diet. So the delay that there is between when they're eating and the response that they have for the antibody drug, it is like slight delay. So the interpretation for that is that if the negative happened on that category, it's because it increases the antibody drug when compared with the hay that is they're being well fed. It's a little bit of a mouthful to do, and it was a little bit of a mouthful also when I coded it, because we have that issue, but also the nutrition that comes from the antibody, the population level nutrition, has that specific delay with respect of the individual level nutrition. That's why I look at both the fecal chlorophyll and the population level one.
And then the second question is kind of building off that, but like also maybe forecasting of what you might anticipate going into the future with these results of with impacts of climate change. What do you anticipate seeing in the population dynamics of the disease? Do you think more individuals become infected and then therefore cause more potential spillover, or not spillover in the sense of like between the cattle and the buffalo?
So the answer to that is yes. I would expect some changes, because just any type of change on the environment, right? It might modify migration patterns, for example. So that changes the interaction that they have with cattle, for example, or even the interactions within themselves, right? If climate gets warmer, right, then there are more droughts, then they would have like that situation where their antibody protection will drop more likely because they are well fed, they're ill fed. Or the interaction with more individuals, they just keep dragging to points where they are actually having the food, right? Foraging strategies of like during a drought, they're going to be meeting more often. They're really social individuals, so there are specific hotspots where they're going to be facing, if conditions changes on their environment. Although I did not include that type of environmental conditions on this one, so that's a good question. Yes.
I know you are not sharing your results on chapter 3, but would you mind sharing like what is the main result that you got from the evolution of the pathogen the model itself?
Sure, I can give you like an example. So the general idea for this model is that susceptible individuals are, they're considered susceptible with some immune memory. So if you have no immune memory, you're a completely naive individual, you have never been infected whatsoever. After a certain amount of time, you are susceptible with some immune memory, but maybe not for all different strains. Those individuals, they can be infected with a strain that is differently enough to the one that you have been infected before. And then you can become susceptible again or recover with an improved immune memory. Looking at just having immune memory for the most recent infection. So say I have 10 strains of serotype 1. I got infected with strain 5, and then I'm recovered from it, but I'm still not immune for all the other 9. I can still get infected by the others. Now the pathogen is also evolving, right? So in the environment, if strain 5 was present and eventually they changed to a different enough strain, I might be reinfected again, right? So that type of behavior is the one that we were expecting. In this graph, basically we have a high density of the initial infected population of a specific strain. As the pathogen evolves, new strains keep emerging. They recover individuals from, with that immune memory of a specific strain. They just keep catching up, but then, new pathogens keep emerging for those other ones, and then they just keep, getting recovered, while the completely naive population eventually just dies out, right? So then, at the end, after a long period of time, you have a set of challenges between the recovers with the different strains and the newer strains that are passing. And then the idea is that you're looking at almost a predator-prey type behavior between your immune system and the pathogen. But the pathogen keeps changing, so it keeps adapting to the tools that the predator is having. So you always have that mass dynamic, or the red ring hypothesis that it usually is shown in the processes. So that's basically what it is. And
I think we need to wrap it up and go to the rest of the event.
Yep.
But you can ask shop questions when we all gather for food and drink. Ricardo will love it.